Today we are going to discuss about eye cell disease, a very important topic for your exam. We are going to divide this lecture into several parts. In the first part, we are going to discuss the origin of eye cell disease. In the second part, we are going to learn about its clinical manifestations, laboratory findings and its diagnosis, as well as how do we differentiate eye cell disease from some other disease with which it shares common clinical features, and in the final part is prognosis and treatment options. So eye cell disease is a lysosomal storage disease. Lysosomal storage disease are a group of disorders where one or more lysosomal enzymes are either absent or defective, as a result of which various substance tends to accumulate inside cell. Eye cell disease is a type of lysosomal storage disease which is known as mucolipidosis 2 alpha or beta. So mucolipidosis 2 alpha or beta. The mode of inheritance will be autosomal recessive. Now let's dive into the origin of eye cell disease. We are going to see into its origin from different angles. We have to know the flow of events that occurs in eye cell disease. The underlying defect in eye cell disease is a genetic defect, which in turn leads to an enzyme defect, and which will ultimately lead to a protein targeting defect. So we are going to discuss the genesis of eye cell disease at different levels: genetic level, enzymatic level, as well as protein targeting level. So let's begin with the protein targeting part. Now what is protein targeting? Protein targeting is the journey of a protein from its site of synthesis to its final site of localization. Now there are two pathways of protein targeting. One pathway is known as EOR pathway, and second pathway is known as cytosolic pathway or branch. The term EOR itself denotes that the proteins of EOR pathway has to go through endoplasmic reticulum. So the proteins of EOR pathway first comes to endoplasmic reticulum. From there it has to go to Golgi apparatus, and from there it will reach its final site of localization. What are those organelles? Peroxisome, lysosome, and there is secretory proteins. Whereas the cytosolic branch targets proteins from ribosomes to organelles such as mitochondria, nucleus, and chloroplast. Now this Golgi apparatus is, as you know, is the sorting and packaging center of cell. Sorting and packaging center of a cell. Now Golgi apparatus does this job by identifying certain sorting signal present in the proteins. So for lysosomes, that sorting signal which is attached at Golgi apparatus by some enzyme is mannose 6-phosphate. So mannose 6-phosphate signal, what if the enzyme that adds mannose 6-phosphate becomes defective? In that case, the lysosomal enzymes will fail to reach lysosomes, and instead they will make their way into blood. So blood level will be high, but in lysosome the enzymes will be absent. Because of the enzymes being absent, lysosome will not be able to degrade various type of substance that cells engulf. As a result, this substance will accumulate inside cell. Lysosome will swell, and this will inclusions inside cell. That's why the name of the disease is eye cell disease. So I want to zoom into the Golgi apparatus where certain enzymes act to add this mannose 6-phosphate sorting signal to the lysosomal enzymes. This enzymatic step that adds mannose 6-phosphate is a two-step process. But before I get into that, remember lysosomal enzymes are glycoproteins, so it has a protein part and to which a oligosaccharide chains are attached like antennas. So let's assume this is our lysosomal enzymes. So this is the protein part and to which various oligosaccharide chains are attached like antennas. In this entire structure, let's say this is the specific mannose residue to which a phosphate group must be attached to act as a sorting signal for lysosomal enzymes. Now let's see how this mannose 6-phosphate signal is attached. So I have mentioned that it is a two-step process. The first enzyme that will come and act is known as GNPT. So GNPT stands for N-acetyl glucosamine 1-phosphotransferase. What is its function? So it is transferring N-acetyl glucosamine 1-phosphate from UDP N-acetyl glucosamine to specific mannose residues of lysosomal enzyme. So what is the product that will be formed here? So this will be the product. This is our mannose residue, specific mannose residue to which a phosphate will be attached plus N-acetyl glucosamine. Now the problem here is that here the mannose 6-phosphate sorting signal, so this is our mannose 6-phosphate sorting signal, it is masked by N-acetyl glucosamine residue. So this has to be removed in the second step. Now the second enzyme will come and play, which is known as NAGPA. NAGPA stands for N-acetyl glucosaminidase. So it basically breaks this phosphodiester bond between the phosphate group and the N-acetyl glucosamine, releasing this N-acetyl glucosamine from the structure. Now what we get? We get a product where mannose 6-phosphate is attached to the specific mannose residue. Now it will be targeted to lysosome, where specific mannose 6-phosphate receptors present on lysosomal membrane will recognize this signal and uptake inside it. This is how exactly mannose 6-phosphate is added to lysosomal enzymes. But what happens in ICL disease? Because of some genetic defect, the enzyme GNPT, which is responsible for attachment of N-acetyl glucosamine 1-phosphate to specific mannose residues, is absent. This is the problem with ICL disease. The enzyme GNPT is absent because of some genetic defect. In the next slide, we are going to discuss the genetic defect in itself. So GNPT, N-acetyl glucosamine 1-phosphotransferase, it is a hexameric protein consisting of 2 alpha, 2 beta and 2 gamma chain. The different subunits has different function. Alpha and beta is the catalytic unit and the gamma is recognition. Now out of these 6 subunits, alpha and beta are product of one gene and gamma is a product of another gene. There are 2 genes, one is, we will call one gene as GNPT AB and another is GNPT G. From GNPT AB we get alpha and beta polypeptides and from GNPT G we get the gamma subunits. Now mutations in GNPT AB can be of 2 types. One is null mutation and second is a loss of function type of mutation. In null mutation, there is complete absence of GNPT, and loss of function mutation, there is or deficient function of GNPT. When enzyme is totally absent, the disease is I cell disease, also known as mucolipidosis 2 alpha or beta. And when there is deficient action of GNPT, that is some level of enzyme activity is present, the disease is mucolipidosis 3 alpha or beta, also known as pseudo-Hurler syndrome. This is the classical variant of pseudo-Hurler, which is caused by mutation in GNPT-AB. So mutation in GNPT-AB is responsible for two diseases. One is I cell disease, where there is null mutation, and there is loss of function mutation, where there is enzymes activity present but it is reduced to a large extent, responsible for the disease pseudo-Hurler syndrome. And GNPT-G mutation is also possible, and this will cause another variant of pseudo-Hurler, which is mucolipidosis 3 gamma. Actually, it is also pseudo-Hurler syndrome. By the way, one more thing, mutations in NAGPA gene, the second gene that is causing the uncovering of N-acetyl glucosamine signal, is not known. So there is no disease which is caused by mutation in NAGPA gene, or also known as uncovering enzyme. So no disease has been reported yet. In the next slide, we are going to see the summary of events that are responsible for I cell disease. Mutations in GNPT-AB gene is the starting event, which will result in defect in GNPT enzyme. Enzyme will be absent. Mannose 6-phosphate sorting signal could not be attached to lysosomal enzyme. This leads to faulty targeting of lysosomal enzymes. They will be absent in lysosomes, but their level will increase in blood and urine, as a result of which in cells there will be accumulation of various storage materials like cholesterol, phospholipids, sphingolipids, and this will lead to formation of phase 10's inclusion bodies will be found inside cells. So this is the summary. What are the laboratory findings? First, lysosomal enzymes will be absent or even largely reduced inside cells. Increased level of enzymes present in plasma, because the lysosomal enzymes could not reach the lysosomes, they will make their way into blood. The third laboratory finding is the formation of various type of inclusions, and there will be a striking finding that the glycosaminoglycans levels are not increased, and their urinary excretion of glycosaminoglycans are more or less normal. Okay, so the diagnosis of I cell disease, because the clinical features that I cell disease presents with are non-specific and can be found in many other lysosomal storage disease, so we cannot rely on clinical presentation to make a diagnosis. What is important here is the biochemical diagnosis. We can theoretically also do enzymatic assay by measuring the level of GNPT enzyme. So we can do GNPT assay, and genetically we can also try to detect the mutations in the affected gene like GNPT-AB gene. Mostly we rely on biochemical diagnosis. I cell disease presents with severe psychomotor retardation, delayed developmental milestones, short stature, typical quartz facies, skeletal deformities, craniofacial abnormalities, presence of cardiomegaly, hepatomegaly, umbilical hernia, corneal clouding, and gingival hyperplasia. The cause of death could be due to congestive cardiac failure or recurrent upper respiratory tract infections. So how do we differentiate I cell disease from pseudo-Hurler syndrome? If you remember from our previous discussions, so I cell disease was mucolipidosis 2 alpha beta and pseudo-Hurler was mucolipidosis 3 alpha beta and gamma. Both of these diseases have the common underlying defect, that is a faulty targeting of proteins to lysosomes due to mutation in GNPT gene. As you can assume, there will be not much difference clinically between these two disease entities. So how do we differentiate? We can differentiate on the level of severity. Eye cell disease is caused by null mutations in GNPT-AB gene. The presentation will be much more severe and an early manifestations compared to pseudo-Hurler syndrome, which is due to a loss of function mutation. So the presentations are less severe and even a late presentation, and life expectancy is also higher compared to eye cell disease. Patients with pseudo-Hurler often survives until adulthood, whereas eye cell disease patients mostly die in their first decade of life. This is how we can differentiate eye cell disease from pseudo-Hurler syndrome. Comparison with Hurler syndrome. So Hurler syndrome is not a mucolipidosis, it is a mucopolysaccharidosis. So it is MPS type 1. Can anyone name me the enzyme that is defective in MPS 1? So it is L-alpha iduronidase. So this is the enzyme that is defective, and because of this enzyme defect there is accumulation of glycosaminoglycan such as dermatan sulfate, heparan sulfate. So these materials will accumulate in blood and urine. There is increased excretion of glycosaminoglycans in urine, or there is presence of mucopolysaccharidoria. This is a very characteristic finding of Hurler syndrome, by which we can differentiate it from eye cell disease, because in eye cell disease urinary glycosaminoglycan excretion is normal. So remember this point, in eye cell disease urinary GAG is normal. In Hurler syndrome, increased excretion of glycosaminoglycan in urine. So by this method we can differentiate eye cell disease from Hurler syndrome. Prognostically, as there is no curative treatment available, the life expectancy of this disease is between 5 to 8 years. Most of the child dies because of congestive cardiac failure or recurrent upper respiratory tract infections. Treatment-wise, we have not much options. Mainstay of treatment include physiotherapy, surgery and bisphosphonate therapy. Physiotherapy helps to alleviate pain to a great extent. Surgery may be needed, such as knee or hip replacement or valve replacement therapy, depending on case-to-case basis. And bisphosphonate therapy by inhibiting osteoclast helps to relieve pain. Now there are also some experimental treatment options available, but remember these options are only being tried on animals and they are yet to make their way to be used on humans. So genetic therapy, enzyme replacement therapy, hematopoietic stem cell transplantation, antisense oligonucleotide and pharmacological chaperones. Just remember these names. Okay, so we are done with today's lecture. I hope you have learned something new, and if you have questions regarding today's discussion, feel free to ask them in the comment section. I will be glad to answer them. Thank you for watching.